All right, guys, welcome back. So let me try and get out my pointer here. So remember that now in our activities through the Galaxy server, we've used our next generation sequencing aligner. Right, so we chose to use Bowtie 2. Remember, at every step of this pipeline, there are a number of different software packages that we can choose to use. So our aligner moved our FastQ, or our kind of raw output from the next generation sequencer, to what's called the compressed version of that sequence alignment map file, the SAM file, to the BAM file. We then used Freebase, which is a well-known variant caller, to determine where our sample sequencing reads that we got from our next generation sequencer differed from the human reference, and that took as input our BAM files and output what's called a variant call file that has a very a certain type of structure that we expect to see, that we went over in the previous online lecture section. And now the next step of our process is to annotate those variants. And so a list of variants, detected in a set of samples is a very wonderful start. But to discover, clinically relevant or biologically relevant, in the case of research, it's impossible without, some additional tools and data. And so in particular, what we use variant annotation for is to kind of prioritize that list of variants, because you can imagine that any number of sequencing reads or sequencing or clinical samples will differ from our reference genome in millions of different places, and not all of those are relevant to the particular trait that we're interested, the particular disease we're interested. So we want to be able to prioritize, those specific base pair changes that which could possibly lead to our clinical outcome. And so this allows us to prioritize our variants. It allows us to use specific filtering mechanisms. This is often based on, inheritance patterns that are expected for, a particular disease-causing variant. So remember, oftentimes we're looking for Mendelian disease, and so we know that if they have, one or possibly two copies of a very rare allele in a gene that could cause that disorder, that they are very likely to display the disease. And so we're kind of we're trying to drill down to that one variant that we can show may be causing that particular disease. And then obviously we want this reported in a very human-friendly way, so a way that we can read it and interpret it. And so we decided to use a program called SNP-F. And again, if you want to read the research paper, it's here. And this piece of software annotates and predicts the effects of genetic variants or genetic changes from our reference. And so here's what we have. We decided to use SNP-F. F, remember, is the program that you go to within the Galaxy server to submit all of your jobs. And here's our proposed pipeline from beginning to end. So we took our FastQ files from our NGS sequencer. We used Bowtie 2 to align those FastQ files to the reference and produce our BAM file. We took our BAM file, we performed variant calling on that BAM file to produce our variant call file, and then we annotated our VCF using SNP-F. And now our goal is to filter those variants to determine the genetic variant that's most likely causing our specific disorder. And if we remember back to our activity, um, we had what's called a genetic trio in medicine. So we have a father, a mother, and an affected son. And so this particular child, oftentimes called the proband within the genetic trio, is affected with a disorder called osteopetrosis. And you can look up osteopetrosis, but basically it's a bone growth disorder that causes very dense formation of the bones. And this actually leads to, you know, more prone to fractures and breaks and other things. And basically our job in this particular activity that we went through is to discover the genetic variant that may be causing, this particular child to express that particular phenotype and where he actually inherited that particular disorder from the mother or the father. And so remember that when we were going through our VCF structure, that all VCF annotations that we run get kind of thrown into the VCF info column, right? And so you might be asking, what are some important annotations that we can perform, on our VCF file that we got from the Freebase program? And so we can do things like gene-based annotations. So is the change within an exon of a gene? And so with the whole exome sequencing, we're going to get some overlap with intronic sequences or sequences that aren't immediately translated to proteins. And those genetic changes in the intronic sequence might be relevant. But it's very difficult to tell because it's not immediately translated to proteins. We can annotate for frequency invariants in existing whole genome exome data, right? So how common is it in the general population? And so remember, with the Human Genome Project and kind of moving on from the Human Genome Project, one of the main kind of deliverables there was to make all of this data publicly available in large databases. And so the Human Genome Project is completely publicly available. Every sequencing, endeavor since then has been put up and is publicly available in databases, and anyone can access that information. And so we have a lot of access at our disposal that we can now use to be able to sift through specific variants and see if they're relevant to disease. We can perform things like functional prediction in our exome data, right? So will the change alter the function of the protein? And so we base this on a number of things. So we base it on, what we know about the side chains of those amino acids, how they actually behave, how they cause the protein to fold differently, what the function of that protein is, a number of things to kind of functionally predict, how a change at that particular nucleotide base will impact the amino acid that's eventually produced. We can also look at things like splice variants. And so, especially in our genomes, the human genome, there are a number of different what are called protein isoforms, which arise from differential splicing. And so we splice in and out certain exons to produce proteins that have very different phenotypes and functions. And so if there is a genetic change where that splice occurs, or where the cellular machinery kind of latches on to cut in and out that intronic sequence, then there could be quite an issue with producing a functional protein. We can also look for things like disease-specific variants. And so along with getting that information into databases, there are also databases of clinically relevant variants that have been found in previous studies that we can also look at. And so we can use all of this information to basically filter. We can basically use at every step of our annotation or what's in our info column, we can use this as filtering criteria for our whole exome VCF. And so our top two, actually, we can perform within SNPF itself. And so I actually did some additional filtering for you and actually gave you a filtered VCF in our case that had relevant variants already filtered for you. You can see a couple of options within our Galaxy server. We can remove, common variation, and so we often do this with a very well-known web-based tool called Nomad, or Genome Aggregation Database. This has about 123,136 exome sequences from the general population and about 15,500 whole genome sequences. And these are all of unrelated individuals. Presumably they should all be healthy. And so we can use this if we're trying to identify, a very rare variant, for example, in a patient, what variants to screen out or keep in our list of possible disease-causing variants. And so remember, I actually had you look up a specific genetic variant in the Nomad database, kind of look at the allele frequencies that it reports for males and females. So that's a portion of our activity that we did shortly before the break. We can also filter based on variant prediction. We didn't actually do this within the class itself, but remember there are a number of tools that we can use. Some of the most popular are called SIFT and polyphen2. And remember, as I mentioned before, these predict whether an amino acid substitution affects the function of that particular protein. An important tool that we can use, especially if we know the inheritance pattern of the disease that we're looking at, is we can filter on specific inheritance patterns that we identify, especially if we have information from a genetic trio like we have in our case. And so osteopetrosis manifests as what's called a recessive disorder. And so remember, or if you don't remember, recessive type disorders, you need 2 copies of the disease gene to express that particular phenotype. And in the case of recessive based disorders, that would mean that if the mother and father are unaffected or do not have the particular disorder that we're interested in, they must be carriers, right? Because they have a 50% chance of passing on that disease variant to the proband or the child in our case, right? So in our case, the particular BAM files that we happen to be looking at, we know the parents are unaffected in our little hypothetical scenario, but the proband has been clinically diagnosed with osteopetrosis. So we can take this information and, we can basically identify, the specific pattern that we expect from our disease-causing variant. And so in this case, we would expect in our VCF file, so remember how our genotypes are termed in our VCF file, so they're broken down into 0s and 1s, 0s being agreeing with the reference and 1s being agreeing with our alternate allele. And so we would expect that our carrier father that's unaffected has a 0-1, right, being heterozygous, and our unaffected mother, also being a carrier, also has a 0-1. Whereas our affected proband, he has the clinical disorder, will have a 1-1, right? So 2 copies of the defective allele.